Coronaviruses are a subfamily of relatively spherical particles with the taxonomic classification Orthocoronavirinae. They have spike proteins coming out from all directions of which they are known for. Spikes give the virus a pointy, crown-shaped appearance which led to its name corona, Latin for crown or wreath. Most particles are in the size range of 80 to 120 nanometers, but some can be as small as 50 nanometers or as large as 200. The virus contains genetic material in the form of RNA, which is a single-stranded polymer made from the nucleotides adenine, guanine, cytosine, and uracil. This genetic material is protected by a nucleocapsid, which itself is enclosed in a lipid bilayer envelope embedded with proteins. The viral envelope is made up of a lipid bilayer, in which the membrane envelope and spike structural proteins are anchored. The membrane and envelope proteins are responsible for maintaining its shape and size. On average, a single coronavirus particle has 74 spikes. The spikes are each about 20 nanometers long and composed of a trimer of the S protein. A trimer is a polymer with exactly 3 monomers bonded together. The S protein is made up of an S1 and S2 subunit. The homotrimeric S protein is a class I fusion protein which mediates receptor binding with the host cell. The S1 subunit has the receptor binding domain, while the S2 subunit forms a stem which anchors a spike in the viral envelope, and on protease activation enables fusion. Both subunits remain linked non-covalently until they attach to the host cell membrane. The S1 proteins are the most important for infection. They possess 2 major domains known as the N-terminal domain, or S1-NTD, and the C-terminal domain, or S1-CTD, both of which serve as receptor binding domains, which means they both get attracted to the receptors on the host cell. NTDs recognize and bind sugars on the surface of the host cell, while CTDs recognize protein receptors such as angiotensin converting enzyme 2 or ACE2, aminopeptidase or APN, and dipeptidylpeptidase 4 or DPP4. When compared to viruses, cells are huge and complex, and they perform many functions. Viruses are simply made up of bundles of RNA surrounded by a membrane with tools such as spike proteins for infection. When viruses are outside of a host cell, they are known as virions instead, because they are considered not alive. Virions are inert particles which move around through diffusion by bouncing off of other virions and viruses. Coronaviruses mainly target epithelial cells and are transmitted from one host to another. Human coronaviruses mainly infect epithelial cells in the respiratory tract, causing diseases like severe acute respiratory syndrome or SARS and COVID-19, while animal coronaviruses mainly infect cells in the digestive tract. The coronavirus gets tethered to cells in the respiratory tract by attaching to heparin sulfate proteoglycan, or HSPG, which is a transmembrane protein on the host cell. HSPG brings one of the coronavirus's spike proteins close enough to bind to a type I transmembrane protein, ACE2. Many of the proteins responsible for catching the virus all have normal jobs. For example, HSPG works to form new blood vessels, and ACE2 regulates blood pressure. Once the spike protein is docked to the ACE2 receptor, a protease like one from the cathepsin family or transmembrane protease serine 2 or TMPRSS2 of the host cell cleaves and activates a receptor-attached spike protein, detaching it from the viral membrane. This leaves space for a class I fusion protein to propel forward from the virus and pierce into the cell membrane. It then coils itself, bringing the virus close enough to the cell to fuse. This allows the virus to go through endocytosis, inserting itself and its contents inside the cell. On entry into the host cell, the virus is uncoated and its genome enters the cell cytoplasm. The coronavirus RNA genome has a 5' methylated cap and a 3' polyadenylated tail, which allows it to act like a messenger RNA and be translated by the host cell's ribosomes. Many non-structural proteins coalesce to form a multi-protein complex. One of the many proteins in the complex directly responsible for replication and transcription of RNA, is RNA-dependent RNA polymerase, or RDRP. The first main function of the complex is to replicate the viral genome. The second main function is to transcribe the viral genome, and the third main function is genetic recombination when at least two of the viral genomes are present in the same host cell. Genetic recombination is the driving force for determining genetic variability within a coronavirus species, the capability of jumping from one host to another. This can be seen with SARS-CoV-2. The original strain originated from Wuhan, China, and eventually evolved into the B.1.1.7 variant from the UK, the B.1.1.351 variant from South Africa, and the P.1 variant from Brazil, many of which evolved about a year after the index case. These transcripted mRNAs are translated by the host ribosomes into the structural proteins as well as the accessory proteins. RNA translation occurs inside the endoplasmic reticulum. The proteins move along the secretory pathway into the Golgi intermediate compartment, which mediates traffic between the endoplasmic reticulum and Golgi apparatus. The RNA is enclosed by the proteins that are assembled together and binded to the nucleocapsid. Progeny viruses are released from the host cell through secretory vesicles by exocytosis. This enables the virus to infect other cells. Out of the different types of coronavirus, the only strain that has a vaccine is SARS-CoV-2, which causes COVID-19. This is due to the extended length of the current pandemic compared to outbreaks of other strains like SARS-CoV and MERS, or Middle Eastern Respiratory Syndrome. The most commonly administered vaccines throughout the world against COVID-19 are the Pfizer-BioNTech, Johnson & Johnson, Moderna, Sputnik V, Oxford AstraZeneca, and Novavax vaccines. Currently, only the first three have been approved and authorized for use. There are three main ways that these vaccines for COVID-19 work. They are RNA vaccines, viral vector vaccines, and inactivated vaccines. The RNA vaccine is a new concept that is known for its use in the Pfizer-BioNTech and Moderna vaccines. The RNA vaccines are composed of nucleoside-modified mRNA, or modRNA, encoding a mutated form of the spike protein of SARS-CoV-2, which is then encapsulated in lipid nanoparticles. Viral vector vaccines use a different virus as a vector which delivers a gene that instructs the cell to make a SARS-CoV-2 antigen, which triggers the immune response. Both of these vaccines don't have a threat of causing illness as they do not use the actual virus particles to induce an immune response. The conventional method of using vaccines is through an inactivated or weakened version of the virus. This is how most vaccines are made, and they work by weakening or killing the virus to the point where it is recognizable and can induce an immune response, but not actually posing much of a threat. Millions have died and the entire world has been negatively impacted by the presence of the SARS-CoV-2 and its spread for the past 15 months. As of March 6th, over 300 million doses of COVID vaccines have been administered worldwide. The demand for vaccines vastly exceeds the supply as almost everyone in the world wants to be vaccinated, yet only 7 or 8 billion people are being vaccinated every day. While the manufacture and distribution are occurring faster than ever, the only way to end this pandemic is to push the limits by investing in maximizing the rate of vaccination. Thank you.